Okay, before we begin, are there any questions?
Yeah, so where exactly are we submitting our number for the infectious disease report? Are we submitting it under the chapter or oh my gosh, the reports where it says submit in a Word doc?
You're supposed to put it in let me show you.
Okay, thank you.
You should be seeing my screen now.
Yes.
So scroll down to week 5 lab. And in the unknown notes, you should be submitting your unknown number. You'll have a button here for submit on the right, or it could be down here. I don't have that because I'm a teacher. I've got a grader button. Any questions about that?
Okay, so we are just submitting in a PD or a Word document then. It's not like commenting because I thought there was something about commenting.
I said, do not add a comment. The comment is for me.
Oh,
And
okay.
it'll be on the right here.
Thank you.
Okay. So I did send out the unknown project, and you guys have gotten started on the gelatin test. You'll get the results to the gelatin test, the gram stain, and the PR lactose next Tuesday, probably Tuesday after the lab. All right. Today is February 3rd, and we're supposed to be starting chapter 5, microbial metabolism. There is no lab today, so I will be there for 15 minutes to answer any questions. But after 15 minutes, meaning at 6:45, I'll be logging off.
Sorry, I have another question. Usually we get our previous week lab back so we can correct it by Saturday for a resubmit. Is that going to happen this week?
I'll try and work on that tonight, but I have not graded last week's lab.
Okay.
Scroll that to the top. All right. Any other questions? If not, let's begin with chapter 5, microbial metabolism. How are you hearing me?
Good.
Okay. So know the major goals and the rough outline. Know the terms: catabolism, anabolism, activation energy, an enzyme, an active site to an enzyme, competitive inhibition, non-competitive inhibition, also called allosteric inhibition, redox reactions, what are photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs, fermentation, cyclic and non-cyclic photosynthesis. Somebody was talking, you've still got your microphone on. Could you turn the microphone off? I'm not able to do that unless I stop the share of the screen. Let's see if I can get that. It looks like everybody's muted. So understand what feedback inhibition is, and then understand what aerobic respiration is. We'll talk about it at great length, know its steps, and how it differs from anaerobic respiration. So that's pretty much what we'll be doing. You should know that metabolism is all of the chemical reactions in an organism or in this case, because we're talking about a microorganism, all of the chemical reactions within a cell. We can divide metabolism into two parts, catabolism and anabolism. Catabolism is where you take a complex molecule, meaning a larger molecule such as starch, and then break it into a simple molecule such as glucose. When you break a big molecule into smaller molecules, energy is released. You always leave energy to heat, and some energy may be able to be captured and then be used for building ATP. Now, this diagram is a little bit of an oversimplification because it's showing you catabolism as generating ATP. Human cells are very lousy at generating ATP. There are only two chemical reactions that we can do to generate ATP. And we'll talk about each of those two in this lesson. The first is aerobic respiration, which is a number of steps. But whenever our cells engage in aerobic respiration, we digest larger molecules, making smaller molecules in catabolism. And in the way, we can generate ATP. The other way that our cells can do this is from a fermentation reaction. And we'll talk about that. Now, green plants are better at generating ATP from different catabolic reactions. However, they only have a few catabolic reactions to generate ATP, and they have many catabolic reactions which do not generate ATP. So do you understand my point? And that is, there are many catabolic reactions, only a few, and in humans, only two, can generate ATP. The rest of the catabolic reactions do not generate ATP. Catabolism is a decomposition reaction where a larger molecule is made into a smaller molecule. Energy is released by this process called catabolism. Anabolism is the other half of metabolism, and that's where you combine simple molecules such as glucose. Energy is taken up by the system, and then we can make a larger complex molecule. Usually energy is donated to anabolism by the breakdown of ATP. So anabolic reactions take smaller molecules and energy to make complex molecules. And we do this to biosynthesize our larger molecules, such as starch, proteins, and lipids. Now, because of the laws of thermodynamics, whenever you convert energy, like in a simple molecule, to another form, like a complex molecule, you will lose some energy as heat, and that's shown here. But this reaction does take in energy, but it also gives off energy in the form of heat. Any question about any of that? You can string different catabolic reactions together to make a metabolic pathway, such as when, let me blow this up a little, molecule A is converted to molecule B by enzyme 1, and then molecule B is converted to molecule C by enzyme 2. Molecule C is converted to molecule D by enzyme 3. Molecule D is converted to molecule E by enzyme 4. And molecule E is converted to molecule F by enzyme 5. You can string all of these reactions together by putting in all of the enzymes. And then you get from molecule A, you'll get as an end product molecule F. Molecule A is called the starting substrate because it's the first molecule in the pathway. All of the other molecules are called intermediates. And molecule B is the product of the first reaction. So we call it an intermediate product. But molecule C is an intermediate, molecule D, molecule E. Molecule F is the last product made. So this one we call the end product. Any question about that? Cells like to make certain molecules, like molecule F, from a starting substrate, such as molecule A, and they will string the reaction together by using different enzymes. And then they'll have a metabolic pathway in the cell to make molecule F. These metabolic pathways are determined by enzymes. And enzymes are encoded by the genes in a cell. Any question about any of that? There's a huge amount of information, chemical reactions going on in the cell. And this graph is trying to show you all of the things that are going on in a cell. I only want you to know most of it, only kidding. Just to understand how much chemical reactions are going on in the cell. Chemical reactions occur because of the collision theory. The collision theory states that chemical reactions occur when atoms, ions, or molecules collide with each other. The reaction occurs when the atoms, ions, or molecules have enough energy to get over the activation energy, the energy needed to disrupt the electronic configurations of the colliding atoms, ions, or molecules, and then has enough energy to change the electronic configuration to make the products. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. The reaction rate can be increased by increasing the abundance of enzymes. More enzymes, we can convert more of the starting substrates into the products. You can also increase the reaction rate by increasing the temperature. When you increase the temperature, the starting reactions have more energy and they will be more likely to hit each other to collide, as well as collide with enough energy to get over the activation energy. Now this one's a little harder, and that is you can increase the reaction rate by increasing the pressure. By increasing the pressure, you increase the chance that the atoms, ions, or molecules will collide with each other. And because they're under higher pressure, they will have more energy, so that if they collide, they'll be more likely to gain the activation energy, which will make it more likely that the collision will convert the starting substrates into the products. Any question about any of that? This graph is showing you the activation energy, and on the y-axis we have the energy level. So down here is, well, down here is 0 energy, and as you go up, more energy. And then this is just time on the y or the x-axis. All reactants have a starting energy, and these energy reactant is a big molecule that's going to break into 2 smaller molecules. And so the starting substrate, meaning A, B, hooked together, has more energy than A and B, the products, when they're separate. An example of that would be something like burning glucose. Glucose burning into carbon dioxide and water. The reactants have more energy than the products, the carbon dioxide and the water. To get from the reactant to the product, we have to go over an energy hump called the reactivation energy, the activation energy. You'll notice that without an enzyme, the reactant has to go over a very large hump. It has to gain a lot of energy before the reactant will get enough energy to convert into the products. And that is called the activation energy without the enzyme. For example, we can convert, let's see, the sugar sucrose. No, that would be the sugar glucose, I guess. The sugar glucose into the products, CO2 and water. However, to do so, we would have to take a blowtorch to it to heat up the glucose and burn it into CO2 and water. And that is why the sucrose has to be heated, has to get a lot of energy. It's probably 1,000 degrees Fahrenheit to get to the activation energy without the enzyme, and then can be converted into the product. Any question about any of that? Well, glucose is happening in your room where it is converting into CO2 and water all the time. And where is glucose being converted into CO2 and water in your room? Here's a hint. You don't need to get a blowtorch to convert that glucose into CO2 and water. Where's that happening?
Did you say in our rooms?
Yeah, in your room. Where is that happening?
Is it in the air?
Well, I was trying to go for in your cells.
Oh.
But it could be if there's bacteria floating around in the air, then the bacteria could be doing it as well. But the point is, in your cells, glucose is being converted into CO2 and water all the time. And how that's happening is because your cells have an enzyme, and the activation energy with the enzyme is much less than the activation energy without the enzyme. As I mentioned, you need to heat the glucose to about 1,000 degrees Fahrenheit to get it to change into CO2 and water. However, in your cells, this is happening at 37 degrees Celsius, which would be 98.6 degrees Fahrenheit, quite a bit cooler. And that's showing you that the activation energy with an enzyme is much less than the activation energy without the enzyme. Any question about any of that?
Since we're getting more energy than we're putting in, is this the reaction that helps us maintain body temperature, or is there something else that helps us maintain body temperature?
Um, you do this to mainly make ATP, but you can do this if you need heat to warm your body, meaning that in warm-blooded animals, some of this heat given off can be used to help warm the body. So the majority of the burning of glucose into CO2 and water in your cells, the majority of that energy is captured and then made to make ATP. And maybe I shouldn't say the majority, I should say the biggest part. But there is a little bit that is used to help keep your body warm. And you always lose some energy by heat. And in warm-blooded animals, we can use that energy to help warm the body. Now, on a hot summer day, which admittedly it's not, but on a hot summer day, that would overheat the body. And then the body would have to spend energy to cool the body. And then you'd start perspiring. Any question about any of that? All right. So we mentioned that how chemical reactions happen in cells is because enzymes perform the chemical reactions. You should know that enzymes are biological catalysts. They speed up biochemical reactions, and they can speed them by 10 to the 8th or 10 to the 10 fold, meaning 10 times 10. You don't need to know this number, but you should know that enzymes are biological catalysts, and enzymes are encoded in your cells by your genes. Generally speaking, you have one specific enzyme to work on one specific substrate and then convert that substrate to a product. Now, as you know, we're going to talk about an enzyme as being specific that way. But as you know, there are some drugs which can mimic the substrate so that, what's my point? That enzymes aren't exactly the specific where they only act on one specific substrate, but essentially it's true. And so we'll talk about it that way just because it's easier to talk about enzymes, that one enzyme is specific for one substrate and it can convert that one substrate to one product. The specificity of the enzymes happens to do with its three-dimensional shape. An enzyme acts specifically on one substrate because the enzyme fits and binds to that one substrate. You should also know that enzymes are not used up or altered in a chemical reaction. That enzymes will actually bind to the substrate and form an enzyme substrate complex. That complex orients the substrate to increase the probability of the reaction occurring. And it puts stress on the bonds of that substrate that help convert the substrate into the product. and this lowers the activation energy. You should know that enzymes normally end in ASE or ACE. And we'll talk about a few of them like transferase, catalase, ligase, but not all enzymes end in ASE. If you happen to know renin, it's an enzyme in your stomach and it curdles milk to help digest the milk. You do not need to know any of these terms. You should just know that most enzymes normally end in ASE. You should also know that enzymes do not work in a vacuum. Some enzymes require a helper in order for the enzyme to function. The helper can be a coenzyme or a cofactor. So there's an enzyme and it gets together with a helper, a coenzyme, which is a biological molecule, or with a cofactor, which is usually an element like iron, zinc, magnesium, or calcium. And then the element gets together with the enzyme to make the finished working complete enzyme. And then the complete enzyme gets together with the substrate. You do not need to know the terms apoenzyme and holoenzyme. You might know the term coenzyme though, because we'll talk about a few coenzymes. And this is trying to show you that the substrate will bind into the complete enzyme. Some important coenzymes are NAD, NAD+, or nicotinamide adenine dinucleotide. Another one is FAD, flavine adenine dinucleotide. You do need to know these two because we're going to talk about them in aerobic respiration. Some other coenzymes are shown here, and you don't need to know NADP+, FMN, or coenzyme A. As I mentioned, let me blow this up a little. An enzyme can fit specifically with the substrate. That an enzyme has an active site to it, which binds directly to the substrate. And then the enzyme and the substrate form a complex that puts stresses on the bonds of the substrate, helping convert the substrate into the product. Once the product is made, the product falls off the enzyme, and then the enzyme is then free again to bind to the substrate. This is known as the lock and key model of an enzyme, where the key, which is the enzyme, specifically fits one lock, the substrate, and then the key can unlock the door or whatever, unlock the lock. Now, this is a simplification of the way enzymes work, but it does tell you that an enzyme specifically binds to one and only one substrate, and that binding is by the active site of the enzyme, the region of the enzyme that binds to the substrate. Now, the lock and key model is a little too simple, and so they came up with the hand in the glove model to explain how an enzyme works. And here we have the enzyme, and there we have the substrate. And that's the active site of the enzyme, which binds to the substrate. What do you notice about the hand in the glove model? What do you notice about the enzyme and the substrate? There's three things that you should understand.
It closed around the substrate. The enzyme did.
Yeah, the enzyme actually changes its shape and then closes around the substrate when they're together in the enzyme substrate complex. So the enzyme slightly changes its shape. Okay, what else do you notice? What do you notice about the size of the substrate and the size of the enzyme?
Enzyme is bigger.
Yeah, the enzyme is much bigger than the substrate. Scroll it over. And the last one, you have to look very closely. Do you notice the substrate? It's a little flatter here compared to there. So in a way, the hand in the glove model doesn't quite work. And that is that the glove will mold to fit the hand. And that's what the enzyme is, changing its shape to fit the substrate. However, it's more like the shapeshifter's hand molding to fit the glove, as the glove is molding to fit the hand. If you know Star Trek: The Next Generation, there was a shapeshifter and he could mold his body to, I don't know, fit like a glove. And the point is the substrate is also changing to mold the hand, to fit the hand. So both the substrate and the enzyme are actually changing their shape to form the enzyme substrate complex. Any question about any of that? All right, there are various factors which can influence the activity of an enzyme. All enzymes work at an optimal temperature, an optimal pH, an optimal salinity. They work generally best without the presence of heavy metals, without alcohol, and without UV radiation. These latter molecules, actually even the earlier molecules, at the wrong temperature, the wrong pH, the wrong salinity, in the presence of heavy metals, in the presence of alcohol and UV radiation, you cause the protein to change its shape and denature. You can also influence the enzyme's activity by varying the substrate concentration. And I'll show you a picture of that. Generally, as you increase the substrate concentration, the activity of the enzyme increases. You can have various poisons like cyanide and fluoride, which will decrease the enzyme's activity. And what's happening is the enzyme poisons start degrading or denaturing the protein, and then eventually stop the protein from working totally. You can also influence the amount of enzyme activity by inhibition. And there's two types of inhibition: competitive inhibition and non-competitive or allosteric inhibition. And then lastly, we'll talk about feedback inhibition, how a cell can use this inhibition to regulate the contents of the cell. All right, any question about any of this? We'll talk about each of these more in greater detail.
Do you know if having like a high blood alcohol level could mess with enzyme activity?
It really would not decrease the enzyme activity in your blood because the alcohol concentration would not be high enough. And to get it high enough, that would be lethal to people. So increasing your blood alcohol content would not increase the alcohol high enough to start degrading the enzymes. Enzymes can be denatured by temperature and pH. Denature is when we have an actional and functional protein, and it has a specific folding pattern as shown here. And then we can denature this protein and have it, I don't know, just looped about here. An example of a denatured protein is egg white when you open up an egg. And then when you heat it, you denature the egg white and make it a solid, which is what we much prefer to eat. Any question about any of that? Why did the protein become denatured at incorrect temperature, pH, or whatever? Why would this protein become denatured if the temperature is incorrect? Nobody has a guess? Well,
because you're
remember
breaking the
that there
bonds?
are different amino acids interacting in a protein. For example, what's one that would be easy to explain? It's kind of complex because it's right here, but we'll talk about right here. The amino acid in this part of the protein and the amino acid in that part of the protein are interacting right there. And it looks like there's probably either a bond, either a sulfhydryl covalent bond, or an ionic bond, or a hydrogen bond being formed right here. And let's say it's a hydrogen bond. If you increase the temperature, you can break that hydrogen bond very easily, and then the protein will denature. And so instead of it being bonded right here, there is no hydrogen bond right there. You could also, by increasing the temperature, there might be repulsion between this amino acid and that amino acid. And you increase the temperature. Now, if that was an ionic bond, how would that change? That wouldn't change. You'd still have repulsion here. Excuse me. So increasing the temperature can break the hydrogen bonds. And it could also, theoretically at least, like maybe put in a hydrogen bond right between here and here, where there isn't supposed to be a hydrogen bond. That's one way you can denature a protein. At the optimal temperature, the enzyme will work at the best rate, have its highest activity. And this enzyme looking like it's going to work best at 37 degrees Celsius. So this could be an enzyme inside our body. And then when we move away from 37 degrees, the amount of enzyme activity decreases. And when we move way away from the optimal temperature, the enzyme is so denatured that it cannot function. For example, if we were to freeze the enzyme, it's not going to work. You'll have no enzyme activity. And then if we were to boil the enzyme, it also would have no activity. The same thing happens with the pH. At the optimal pH, the enzyme has the highest activity. And as you move away from that optimal pH, the enzyme activity decreases. The enzyme starts folding into the wrong shape. And then at extreme pH, pH 0 or pH 10, the enzyme has no activity because the enzyme is denatured, totally denatured. What enzyme do you know of that has an optimal pH around pH 5? What enzyme do you know of that works best around pH 5? Okay, well, pH is 5 is slightly acidic. Where on your body is part of your body slightly acidic?
Stomach?
The stomach is not pH 5. The stomach would be pH 2 or pH 1, but that's a good guess. Your stomach is acidic, and the enzymes in your stomach have to work under acid conditions, but that's very strongly acidic, and the pH gets to about pH 2. Anybody know which is about pH 5? Where in your body is pH 5?
Is it an organ?
It's your skin,
Oh,
which the skin would be considered an organ. It is altogether, it's an organ. Your pH is slightly acidic, and we'll talk about that. So this enzyme is probably an enzyme in your skin. You can influence the amount of enzyme activity by changing the substrate concentration. If the substrate concentration is very low, you don't have very much enzyme activity. The reason being is the substrate is so low that the chance of the substrate binding with the enzyme is not very high. And so the activity of the enzyme is not very great. And as you increase the substrate concentration, you increase the chance that the substrate will meet the enzyme. And so the enzyme activity will increase and will increase until it starts slowing. And then eventually the increase in enzyme activity will not increase at all once you've reached a certain substrate concentration. Why is it that from this point on, when we increase the substrate concentration, the amount of enzyme activity doesn't go any faster? It's actually quite simple. Just think about it. All right, nobody's taking a guess. The reason why the enzyme doesn't increase in its activity rate after we increase the substrate from this concentration is because at this point, all of the enzyme is already bound to a substrate. And if you increase more substrate, you do not increase the enzyme's activity because all of the enzymes are already bound. You cannot increase the binding of the enzyme any further. And that's why the substrate concentration initially increases, and then at a point it slows, and then you increase the substrate concentration, you get no increase the enzyme activity because all the enzyme is already bound. You can influence the amount of enzyme activity by using an inhibitor. And there's two types of inhibitors. A competitive inhibition, where the inhibitor actively competes for the enzyme's active site with the substrate. So here we have the substrate and it binds to the active site of the enzyme. The competitive inhibitor binds to the active site of the enzyme and inhibits the enzyme by competing for the site in the enzyme. So the competitive inhibitor is here, the substrate cannot bind, and then the enzyme will not function. At least this enzyme. Excuse me. There's also non-competitive or allosteric inhibition. This is where an inhibitor acts at a site other than the active site of the enzyme. So here we have an enzyme, and there's its active site. It binds to the substrate to perform the reaction. You can have a non-competitive or allosteric inhibitor, and it binds to the enzyme at the allosteric site, a site other than the active site. And when you have the allosteric inhibitor bound to the enzyme, you change the folding pattern of the enzyme. So the active site can no longer bind to the substrate. Any question about any of that? So the allosteric inhibitor actually changes the shape of the enzyme, and that performs the inhibition. So know how to distinguish between non-competitive inhibition and competitive inhibition. Your body tends to make non-competitive inhibitors, but some of the drugs we take are competitive inhibitors. A cell can influence its enzyme activity by engaging in feedback inhibition. What feedback inhibition does is it regulates the contents of this product, the end product. And if the cell has enough end product, it doesn't want to make any more of this pathway, the pathway to make the end product. And what happens is that substrate can get together with this first enzyme to make intermediate A, which can get together with enzyme 2 to make intermediate B, which can get together with enzyme 3 to make the end product. What the cell can do is, if the cell has enough end product, it can bind the end product to the first enzyme and then use the end product as an allosteric inhibitor to this first enzyme. If you bind the enzyme with an allosteric inhibitor, you change the active site of the enzyme. so it can no longer bind to the enzyme. And then that will shut down the production of intermediate A, which will shut down the production of intermediate B, which will shut down the production of the end product. And so the cell can regulate how much end product is in the cell by using the end product in a feedback inhibition loop, binding to the first enzyme and then shutting it down. Now, cells normally have this feedback inhibition where the end product regulates the first enzyme in the enzymatic pathway to make the end product. Once enough end product is made, the end product then binds to the first enzyme and then shuts it down. Whenever the cell needs more end product, the end product falls off of the enzyme 3. And this falling off is just a stoichiomic reaction. Every once in a while, the end product falls off. And then the enzyme can function. Actually, sorry, it falls off here. And then this enzyme can function. We get intermediate A made, we get intermediate B made, and then we get more end product made. So whenever the cell needs more end product, it can use the feedback inhibition loop to make more end product. And if there isn't enough end product around, then there won't be end product binding to the first enzyme and shutting off the reaction. So the reaction proceeds until we get enough end product made that enough of the end product binds to the first enzyme in an allosteric reaction. And that inhibits the enzyme's ability to bind to the reactant. No reactant binding to the enzyme, then we get no intermediate A, no intermediate B, no end product made. How does the end product fall off of the first enzyme? Well, it's a stoichiomic reaction, so every once in a while, the end product just falls off. If there's lots of end product around, an end product will bind to the enzyme and then shut it down. So the enzyme does fall off every once in a while, or the end product does fall off every once in a while, but if there's lots of end product around, it'll quickly bind to the enzyme and then shut it down. On the other hand, if there's not much end product around and the cell wants more of it, when the end product falls off of the enzyme, there's no further end product to bind to the enzyme. And so we start making the end product again. Any question about feedback inhibition? Here's a concept that is discussed in your books, and I'm just going to talk about it briefly. We have most of our enzymes in our cells as protein enzymes, but there are a few enzymes which are not proteins. These we call ribozymes, RNA enzymes, where the RNA cuts and splices RNA or some other molecule. The RNA enzyme functions as a protein enzyme in that the RNA enzyme has an active site. The active site binds to the substrate. The RNA enzyme is not used up or modified in a chemical reaction, so that once it performs this chemical reaction, it can go ahead and do countless more enzymatic reactions. Any question about any of that? All right, your textbook author, Tutora, does not mention it, But there is one RNA enzyme which we will talk about, and that is the RNA in ribosomes is the enzyme that links the amino acids together on a growing polypeptide chain. Any questions about any of that? All right. If there's no questions about enzymes, let's go ahead and change the topic and talk about metabolic diversity among organisms. All organisms, including microbes, require the following two substances. All organisms need an energy source, and all organisms need a carbon source. Cells need an energy source for the cell to remain alive, but the cell also needs a carbon source. We can base an organism's classification on its energy source. If an organism gets its energy from light, we call this a phototroph, an organism which obtains its energy needs from light. And you all know the green plants are phototrophs. They get their energy needs from light. You should know that some bacteria, all of the cyanobacteria, are phototrophs. Some of the bacteria are phototrophs, where they derive their energy needs from light. The reverse of that is a cell, which is a chemotroph. Chemotrophs derive their energy needs from an inorganic or organic compound. For example, humans are chemotrophs. They derive their energy needs from their diet, largely the solid food they eat and drink. Like if they're drinking Jello, that would be solid while you're eating it, but it would dissolve and become liquid in your gut. Another example of humans being a chemotroph is we digest and consume glucose to make ATP. So we are a chemotroph. You can also divide an organism on where it gets its carbon source. Organisms can be classified as an autotroph if the principal carbon source is CO2. As you know, green plants are autotrophs. They get most of their energy needs from burning glucose or some other molecule. So green plants are an autotroph. And that's because their carbon source is CO2 in the air or in the water. Humans are not autotrophs. We get our carbon source from the things we eat. So we are heterotrophs. We get our carbon from an organic chemical compound, meaning largely glucose. So we are a heterotroph. Any question about any of that? Now, if ever you get confused, go back to the simple classification, and that is divide things on their energy source. If they get their energy needs from light, they're a phototroph. If they get their energy needs from a chemical, they're a chemotroph. If they get their carbon source from light, they are an autotroph. The principal carbon source is CO2. If, on the other hand, they get their carbon from an organic carbon source like glucose, we call these heterotrophs. Humans are heterotrophs. We get our carbon needs from the food we either eat or drink. Now we can consider both the energy source and the carbon source together and then classify organisms based upon their energy source and their carbon source. A photoautotroph is a phototroph, meaning it gets its energy needs from light, and it gets its carbon from a organic chemical source, meaning we humans can get both our energy needs and our carbon needs from the same molecule, glucose or sucrose. That is different for the other trophs. Let me move on to the chemoheterotrophs first because it's easier to discuss. Chemoheterotrophs are chemotrophs. They get their energy needs from a chemical compound, such as glucose or chemosynthetic bacteria, which obtain its energy needs from an electron that it strips out of another atom. And we'll talk a little bit more about that. The chemoheterotrophs are different in that you can have a chemical, organic chemical carbon source, or you can have an inorganic chemical carbon source. The organic chemical carbon source are most of the life forms that you're familiar with, like people, earthworms, E. coli. An organic chemical compound is an organism which gets its energy needs from an inorganic chemical compound, such as sulfur dioxide or hydrogen sulfide. Any question about any of that? Chemoautotrophs are chemotrophs, meaning they get their energy needs from a chemical compound, such as glucose. They're also autotrophs in that they can obtain their carbon source from an organic chemical compound, such as glucose. Well, wait a minute. This is an autotroph that gets its carbon source from CO2 in the air or the water. An example of a chemoautotroph is a chemosynthetic bacteria. It gets its energy needs, or it can get its energy needs from stripping an electron from an inorganic chemical compound, and it gets its carbon source from CO2, either dissolved in the water or the air. Now, the photoheterotrophs are organisms that most of you are probably not familiar with. So you're not terribly familiar with the phototrophs, but they get their energy needs from light. That's why they're called the phototroph. And they get their carbon source from an organic chemical compound such as glucose. An example of a photoheterotroph are the green and purple non-sulfur bacteria. They get their energy needs from light, and they get their carbon source from an organic chemical compound such as glucose. The Photoautotrophs are much easier to understand. They get their energy needs from light, because they're a phototroph, and they get their carbon source from CO2. And that's where green plants are an example of a photoautotroph. But there are green bacteria, meaning photosynthetic bacteria, particularly among the cyanobacteria. And we can break the photoautotrophs into two types, but I'll break that a little bit later. The chemoautotrophs are the chemosynthetic bacteria. They get their energy needs from a chemical compound, and that could be hydrogen sulfide or sulfate, some gas that's toxic to us, or it could be from iron. What happens is the chemosynthetic bacteria strip an electron from the chemotroph, the chemotrophic inorganic chemical compound, and then they use that electron to serve its energy needs. The chemosynthetic bacteria tend to get their carbon from carbon dioxide in the air. And you all know an example of a chemosynthetic bacteria. Just that you don't realize it's probably an iron oxidizing bacteria. If you take up the toilet tank, meaning the place holding the clean water on top of the toilet, and then you flush that to flush the toilet bowl. Well, you know all that brown or yellow muck stuck to the sides of the toilet tank? That's iron oxidizing bacteria. And it's a chemosynthetic bacteria getting its energy needs from iron dissolved in the water, and it gets its carbon dioxide from CO2. Any question about chemoautotrophs?
Do we need to know all of these energy sources and carbon sources?
Yes.
Okay, thank you.
Now the chemoheterotrophs are easy because we're a chemoheterotroph. We get our energy source from a chemical compound such as glucose, and we get our carbon source from a chemical compound such as glucose. With many of the chemoheterotrophs, the molecule supplying the energy source is the same molecule as supplying the carbon source. And for humans, that's largely glucose, although it could be any of the food molecules we eat, because we are a chemoheterotroph. However, there are some bacteria, which like E. coli, are chemoheterotrophs, but there are some bacteria that get their energy source not from an organic chemical compound like glucose, but get their energy from an inorganic chemical compound, such as hydrogen sulfide gas. And if you're wondering, these organisms are a chemosynthetic bacteria. It's just that they get their carbon source from an organic chemical compound, so that the chemosynthetic bacteria are split. About half are chemoautotrophs, and they get their CO2 from the air as carbon dioxide. But half of the chemosynthetic bacteria are chemoheterotrophs, and they get their energy needs from glucose or from an inorganic chemical compound, stripping the electron from something like, an inorganic chemical compound, sulfur or hydrogen sulfide, and then use that electron for its energy needs. But they get their organic or their carbon source from organic chemical compounds such as glucose. So the chemosynthetic bacteria, some of them are chemoautotrophs, others are chemoheterotrophs. Of the animals, all of them are chemoheterotrophs. All right, any question about any of that? If not, let me state you need to know what's in red. You can split organisms by their energy source into chemotrophs, getting their energy needs from a chemical compound, or you can split an organism into a phototroph, getting its energy needs from light. You can also split the phototrophs on where their carbon source is. If they get their carbon from CO2, they're a photoautotroph. But if they get their carbon from an organic chemical compound such as glucose, they are a photoheterotroph. Like I said, you don't really know photoheterotrophs, but the green and purple non-sulfur bacteria are photoheterotrophs. If you get your energy source from a chemical compound, you're a chemotroph. And if you get your carbon source from an organic chemical compound, you're a chemoheterotroph. Humans are chemoheterotrophs, and we get our carbon from an organic chemical compound, and we get our energy needs from a chemical compound. But not all chemotrophs are this way. Some of them are chemoautotrophs, and they get their energy needs from a chemical compound. But the chemoautotrophs get CO2 from CO2 dissolved in the air. All right, you need to know what's in red for the classification of organisms, depending on their energy source and their carbon source. You do not need to know that the photoautotrophs can be split into oxygenic photoautotrophs and then anoxygenic photoautotrophs. You also don't need to know that we have, I lost my train of thought here. Let me see if I can get it back. The chemoheterotrophs can be split into two different groups, and that is that those which get their carbon source from an organic compound, and those which get their carbon source from an inorganic compound. Any question about any of that? All right, I think I'll stop here and we will continue with this next time. So if you have questions, you can show up in the lab at 6:30. If you don't have any questions, then there's no reason to show up. All right, I'll see you next time.
Thank you.
Can we ask a question about the project?
All right, what's your question about the project?
So you are running the gelatin lab for us already, right?
Yes,
And what are the other two ones that are being ran? Sorry.
they're not being run yet. On Sunday, you'll start the gram stain. On Monday, you'll start PR lactose. We'll do those two tests.
Okay. So then when we run the lab, you must send us like a picture of the results, and that's how we try to decipher what it actually is, the bacteria.
Yes.
Okay.
Okay.
All right. Thank you.
All right.